Hi, welcome to the Physionic podcast. My name is Nicholas Verhoeven. I'm a PhD student in molecular medicine and have my master's in exercise physiology, and I've been working in a wet lab, meaning the laboratory that you might see on movies where they're mixing different chemicals and things like that for a number of years. Today's podcast episode, we're going to be discussing a variety of different topics as usual, but I'm going to first address the phospholipase video content that I released just 2 days ago when this releases. And then I'm going to go into some really cool stuff that's coming out of Yale. And then finally, I'm going to end it with a little bit of stuff on antibacterials or antibiotics. So, as usual, there will be timestamps for you if you want to skip, particular topics that you're not that interested in, and you can. But otherwise, you can go all the way through. It should probably be a little bit shorter of an episode this time around. All right, so let's jump into it. A couple days ago, I released a video that looked at a mechanism for muscle growth, muscle hypertrophy. And that mechanism was through an enzyme known as phospholipase D specifically. Now, what the video went into detail with, showing the actual research, showing the data within a particular paper, was that when you have higher expression of phospholipase, that you see greater muscle growth, specifically to the muscle cell exactly. So in an in vitro system, looking at cells specifically. But they did actually do this in mice as well. So that was pretty interesting. Now they also ended up knocking it down, so inhibiting it through drugs, but also through silencing RNAs. Silencing RNAs are essentially molecules that will, silence RNA. They will inhibit genes, really. They will inhibit gene expression, so you don't get the translation of RNA to proteins to actually be functional. So by two different mechanisms, they inhibited phospholipase D, and they found that if you inhibit phospholipase, PLD, then you see decreases in muscle size. So muscle growth is decreased, and atrophy actually is considerably higher. And speaking on atrophy, which is muscle shrinking or shrinking in general, they showed that particular atrophic genes are elevated when you knock down PLD, and they are decreased or inhibited. The genes themselves, the expression of those genes, are inhibited when PLD is overexpressed. So again, it just kind of falls perfectly in line with this idea that phospholipase, this enzyme, leads to increases in muscle growth. But even beyond that, they also wanted to see, what would happen if we ended up adding phosphatidic acid? I keep saying phosphatidic acid. It's phosphatidic acid. It's probably not a big deal, but phosphatidic acid is the product that's formed from phospholipase, and I'll explain that in more detail in just a little bit. But this phosphatidic acid is the downstream effect of phospholipase activity, presumably, and they wanted to see if they didn't manipulate PLD and they just added phosphatidic acid, what would happen? And sure enough, they found the exact same result. So it's not PLD mediated, really, although PLD allows you to produce endogenous within the cell, within your body, phosphatidic acid. So in that regard, it is helpful, but the PLD itself doesn't do anything in terms of binding with some cell growth pattern or mechanism or anything like that. It's going through this phosphatidic acid, or at least that's what this evidence was pointing towards. So it was an interesting paper, definitely really interesting, well done. So That's kind of to catch you up to what that paper was talking about. Now, I know that one of the main questions people then ask me is, well, all right, what can I do to increase my PLD, my phospholipase D? It's not that simple. Like it's really not that simple. I'll have to record some piece of content to discuss why things like questions like that are not that simple. But when it comes to PLD, there's essentially nothing we can do. You're born with a certain amount of PLD or a certain amount of gene expression for PLD, so your muscles are filled with a certain amount, and maybe they might increase it when you exercise, things like that. But for the most part, it's going to be based on just basic patterns, exercising, progressive overload, increasing volume, things like that. That's going to eventually lead to muscle hypertrophy. However, it could be, and this is where the nuance comes in that I hesitate with, it could be that a supplement company could somehow figure out a way to generate phosphatidic acid, because as I mentioned, that leads to the same results. And if somehow that were to be able to be supplied to the muscle cells themselves, and that's the big question, how do you deliver this stuff? I mean, do you inject it? Do you drink it? Do you have to put it in sort of these microvesicles so it's protected, it doesn't get degraded in the bloodstream? I mean, there's a bunch of different things you have to consider. So if you consume, let's say, phosphatidic acid, maybe it just gets degraded and then it's pointless. Like there's really no point in having consumed it. Maybe if you inject it, maybe it only travels a certain distance and then particular enzymes in the bloodstream get rid of it before it can even get 3 centimeters down your into your bloodstream. It's things like that. You have to test that kind of stuff. So when a supplement company says, oh, well, we came up with this molecule that's highly effective, I mean, sure, if they cite a paper where it's in vitro, just like I just did here with this paper, I mean, that's fantastic when you get to just literally dump the molecule right onto the cells. But your body isn't just muscle cells on a plate. It's made up of a lot of other things that it's a physiological system. So things like that are certainly things to consider. Another example of this, actually, that I was thinking about is collagen. So I've looked at a few collagen papers, and I even presented on a collagen paper. I think it was an S&P video or piece of content. And with collagen, when you add collagen to in vitro cells, then that leads to greater production of collagen when they specifically add procollagen. So an immature form of collagen, then you see more collagen production. Again, that's great, and it obviously proves that the cells can produce collagen out of procollagen when supplied with the ingredients. That's fantastic. However, again, it's not an in vivo system. It's not a physiological system. Therefore, you can't immediately say that, oh, well, if I just start supplementing with collagen, therefore it's going to reach my cells, it's going to get integrated, and then they're going to spit out fully formed collagen fibers. You can't necessarily say that. Same thing with phosphatidic acid. I mean, they clearly prove that phosphatidic acid increases muscle cell size, muscle hypertrophy. However, if you were to consume it, would you necessarily get that result? Because there's so many different factors that might get in the way. So it's something that's probably going to need to be researched. Maybe it has been already. I don't know. And it just adds nuance to the whole situation. But anyway, so I wanted to talk a little more detail on phospholipase. There's a paper in Nature, which is probably, one of the big 3 when it comes to journals in terms of in terms of being a reputable paper. It's way up there. And this research group wanted to find out, how does phospholipase work in general? And they essentially characterized how it functions, which is kind of cool. It's a different mechanism by what a lot of other enzymes function, or at least that I'm familiar with, although I'm sure there are many, many enzymes that work this way. But they wanted to find out, well, okay, how does phospholipase that's integrated into the sarcolemma being the cell membrane of muscle cells specifically, that's why it's called sarcolemma, and it's integrated on the inside of the sarcolemma, so on the inside of the cell. And so how does it actually interact with its reactant? So you need a reactant, and then that gets converted to a product. And it's almost like exchanging money for a goods or service. You can almost think of it that way. So the reactant in this situation is phosphatidylcholine. And what PLD does is it takes phosphatidylcholine and it cleaves through what's called a hydrolysis reaction. So it uses water and it cleaves the choline off of the phosphatidylcholine, and that eventually leads to the production of phosphatidic acid. So then with the production of phosphatidic acid, that then directly binds mTOR, which is something I talked about in that content as well, mammalian target of rapamycin, also known as mechanistic target of rapamycin. They keep changing the names. It's mTOR. It's going to stay mTOR, so I'm just going to call it mTOR. And mTOR then is activated or excited to a point where it has all these different effects. That's why it's kind of called the master regulator of growth. And it starts increasing protein synthesis, and it starts decreasing atrophy, all these different things. I'm not going to go into all the different mechanisms, autophagy, the proteasome gets inhibited, protein synthesis, there's a lot of things that go on. But needless to say, just the bottom line is you just needed to know that mTOR is excited, and therefore you see greater growth, or you get growth signaling, I should say. And that's all really interesting. However, how does it do that? How does PLD, you have to ask yourself, how does PLD actually react with phosphatidylcholine? Phosphatidylcholine just bumps into it, and then suddenly it creates phosphatidic acid, and then mTOR just gets excited? No, there are signals. Something is signaling, right? What is that something? That something is stretch. And this is something that a lot of research has been done on now, especially over the last 10, 15 years. Mechanosensation. How do you sense stretch within a cell, like one cell? How does it sense stretch? And that comes down to, let's say you're lifting some weight, right? It doesn't matter if it's an eccentric or concentric motion or contraction, which, by the way, I have some really cool content coming out on exactly that, eccentric versus concentric contractions for muscle growth. That's a little teaser if you've made it, about 13 minutes into the podcast. So anyways, it doesn't matter what kind of contraction. You just get some sort of stretch in the musculature. Let's say it's an eccentric contraction, so it's lengthening. So the muscle cell is pulling apart. And as it's pulling apart, the phospholipase has to move within that sarcolemma. So it actually becomes more exposed to the inside of the cell, because the phospholipid bilayer, the membrane, the cell membrane, which is made up of phospholipids, that's why I called it that, it moves apart because it needs to fill that space that as it's elongating, as it's being pulled on both ends of the muscle cell. So phospholipase is then exposed to the inside of the cell, and then from here it can bind this phosphatidic acid. But what's really interesting is they're congregated around what are called Z-discs. So Z-discs are the sort of defining features of a sarcomere. I know I'm throwing a few words at you here. I would very much encourage that you check out look up a picture. Just literally type in sarcomere, SARCOMERE. sarcomere, and you'll find it. It's really easy. And you have a bunch of sequential boxes, essentially, which are sarcomeres. They're all kind of collected together in a line, and that's what makes up the length of the muscle fiber or muscle cell, the same thing. And so as this thing is, so each end of the sarcomere is made up of a Z-disc. And Z-disc is kind of a line. It's also called a Z-line. Z-line that anchors a bunch of contractile proteins. I'm not going to go into those contractile proteins, but it makes sense to think why phospholipase D would be preferentially located on that Z-line. Because if you're sensing stretch, if you're getting pulled, you want that area where that's really getting pulled to have the most mechanosensation. So that's what occurs. So you get this pulling sensation, and the cell is translating that by opening phospholipase to more of its reactant. But not only that, as it's pulling, there are these areas in the cell membrane called lipid rafts. And they almost look like rafts. That's why they're called lipid rafts, but they're made up of lipids. They're made up of fats, phospholipids, right? And these lipid rafts are primarily made up of cholesterol and a series of other fats. And they're very thick. They're a little bit different from the rest of the cell membrane. And this phospholipase D is anchored within that region. And as the cell is being stretched out, the enzyme is being pulled towards one direction or another direction. And it is actually being pulled towards its reactant, which is also found within, and maybe it's also free floating, I don't know this, but it is found in the cell membrane. So when it's inert, when you're not moving, phospholipase is disconnected. It's like at a distance, like if it had eyes, it could see its reactant, but it can't reach it because it's stuck in its place. And then suddenly it gets pulled towards it as we stretch and as we contract our musculature, and then it reaches just enough to be able to attach to it and convert it to phosphatidic acid and producing that phosphatidic chain that I was talking about with mTOR. So really, really cool. Gotta admit, like how the cell is able to be figured out in a way that allows for this contraction to be translated within the cell to this one enzyme, which has a powerful mechanotransduction potential or ability. And it's inert when we don't move. And the moment that we create some stretch on that cell, it allows that freedom for that enzyme to do what it's supposed to be doing, producing this phosphatidic acid. So there you go. That was I'm almost 20 minutes in. But it is a really, really cool mechanism, and it's certainly informative. And I was fortunate enough when I wrote my article on muscle hypertrophy, in which I have since updated because I had been basing things off of previous research, and with new research, there were a few things that sort of changed a little bit, not massively. And when I updated it, I added this phospholipase mechanism. But I didn't know that much about it at the time, but now I know considerably more about it based off of this paper and the Nature paper that I was just describing, in terms of how phospholipase goes about producing phosphatidic acid. So real cool. Okay, so the next two topics are going to be relatively quick. This one was really cool. Really cool. So researchers out of Yale are trying to find a way of fighting glioblastoma, which is a brain cancer, and along with some other groups, not just Yale, and they've developed a way, check this out, to use the Ebola virus to fight cancer cells. So what they've done is that they've genetically modified, or they've used a chimera, which is essentially different parts or different gene sequences of the Ebola virus. So they're using genetic editing to create an Ebola virus that is highly dangerous to cancer cells, but oddly enough is not dangerous to regular cells. And the reason why this works, and this is one of the biggest things, I even released a piece of content on this talking about, okay, we can cure cancer, but it doesn't matter. I think that's what I called it. And the reason for that is because how do you deliver the cure? If, like, I work with cancer cells, I don't work on cancer, but I have cancer cells that I use to study particular phenomena, just general cell biology, because they replicate quickly, obviously. And it's easy for me to just take my cancer cells in my dish and just leave them out at night. Just leave them out for like a week. They'll die because nothing's taking care of them. They're running out of resources. They're running out of everything. Their pH drops. I mean, there's all kinds of different things that could go wrong with them. So it's easy to kill them. I could suction the media off of them. I could drop a bunch of drugs on them and kill them. I could, there's all kinds of different things I can do to them. That's not that big of a deal. But when it's inside the body, it's a mixture of healthy cells or normal physiological cells and then cancer cells. So how do you target those cancer cells only? Well, that's been the big problem. Scientists, medicine, I mean, we've known how to kill cancer. That's not an issue at all. Probably figured that out like day one. But how do we deliver that? And in this situation, they're not so much concerned about the delivery. They're modulating their treatment, their cure. I'm not going to say a cure yet. Their treatment with the fact that it's far less aggressive towards healthy cells. So healthy cells would presumably, I think what happens is they're taking advantage of one thing that is different between cancer cells, or a lot of cancer cells, maybe not all cancer cells, likely not all cancer cells, and regular healthy human, or they're all human cells, but regular healthy cells. And that is that cancer cells have difficulty because they've shut down autophagy. In certain respects, I know some people go really crazy about autophagy. Let me just be real clear. First of all, I work in an autophagy lab. We do mitophagy and autophagy. That is what I study in the actual wet lab that I mentioned earlier. But cancer cells sometimes dysregulate or deregulate or reduce autophagy because they don't want things to be cleaned up. They want essentially a mess. It's not that they're thinking about it. It's just a way the genes play out, like in terms of the mutations that occur in the genetic code that lead to this downregulation of autophagy. Now, autophagy doesn't just clean up. See, that's the thing. Autophagy doesn't just clean up broken proteins and kind of recycle proteins and things like that. What it also does is it's an intracellular defense system. So with this intracellular defense system, that allows, if a virus were to get into the cell, which happens, autophagy could deal with it by enveloping it and destroying it. However, as I just mentioned, if cancer cells have a worse autophagy system, it doesn't function as well, and that's one of the predicated reasons why it became a cancer cell, is because it was able to downregulate autophagy. Then that means that this Ebola virus would enter those cells and not be eliminated by autophagy, because it would then kill off the cancer cells. However, when it enters the healthy cells, those cells would have a perfectly normal autophagy system and would be able to deal with the Ebola virus. So it's a bit of a gamble, but if they can get this to work, that would be crazy cool. So freaking cool. So I actually titled it for Instagram, because I'm going to be telling a little bit more on this when this releases. I titled it "Fighting Evil with Evil," because I think that's pretty fitting. The Ebola virus, which gave us a scare just a few years ago, and cancer, which, well, scares the living hell out of us every single day. Okay, so that was too cool. The next thing, the final thing that I wanted to briefly touch on was you've likely heard that antibiotics are becoming less and less effective because of, well, all kinds of reasons. People using antibacterial soaps, using you guys can, hopefully, I'm assuming you guys can probably hear this racket right here. It's right outside my window. Anyway, with antibiotics, that's starting to come to a point where you're getting a lot of bacteria that are resistant to antibiotics. And that is for a variety of reasons. One, because of washing hands, right? We use a lot of antibacterial soaps, which we probably shouldn't. Another aspect is that people don't necessarily take all their medicine. They feel like their symptoms are gone, and they've been prescribed medicine for, let's say, 14 days, but they start feeling better after 4 days, and then they feel really perfectly fine after 7 days. Well, you should still take those extra 7 days because there are instances where bacteria can mutate and because of the antibacterial, which will then make them more resistant to that antibacterial. And then if it does come back, which it could easily come back literally like a couple days later, you will have the same disease, same infection, but it will be more aggressive because the doctor won't be able to prescribe you the same antibiotic. It's not going to work as well. It might not work at all. So there are a number of reasons why antibiotics aren't working, and there are a number of different ways that antibiotics do stop bacteria. One is by breaking down the cell wall. Bacteria have a cell membrane, and then they have a cell wall on top of that cell membrane. We, as eukaryotes, have a cell membrane, but we don't have a cell wall. So if the antibacterial will punch holes in that cell membrane, it can inhibit protein synthesis, it can inhibit nucleic acid synthesis, and it can also punch holes in the cell membrane itself through things like polymycins. And it can also have different effects on metabolism. So there are a number of different ways that antibiotics function, but what's really interesting about this particular antibiotic, I guess I didn't really mention this, but a new antibiotic's been designed, and it's a really clever system of how it goes about doing this. This is out of research from McMaster University, and of course I'll have everything linked so you can check it out for yourself. But what this antibiotic does is it blocks bacteria from being able to replicate. Now, it does this through a really unique and creative mechanism. So obviously, if let's say you're exposed to, 10,000 bacteria. They're all the same bacteria. You're exposed to it. It's enough of an infective dose. I'm just making up a number. If it's enough of an infective dose that it enters your body, and wherever it enters, it starts replicating to the point where suddenly 10,000 is 20,000, 30, it just keeps growing and growing and growing, and you have this infection. That's a problem, clearly. So, and it's not necessarily the bacteria itself, although that is certainly a huge factor, but if you have only 100 of that bacteria, your body's not even going to really react to that. You're not even going to feel it. It's not going to matter. But if you have 1.6 million of that bacteria, that's a problem. It's like having a massive army. You have a massive army of 5 million, and there's another army of 12 that are going up against your massive army. You're not going to be that worried about it. But if that 12 suddenly turns into 12 million, you're going to be concerned. And that's essentially what I'm trying to get across here. So what they're trying to do is block this antibiotic blocks bacteria from being able to replicate and produce more of themselves within the body, which will then lead to this widespread infection. Now, how it does that, though, is what's really cool, is that instead of punching holes in the cell membrane or the cell wall, what it does is it locks the cell wall. So because you can imagine if you're going to spontaneously produce another cell out of a cell, that means that original cell has to split into two. So how does it do that? Well, it needs everything to replicate into two, and that includes the cell wall. So that includes the cell wall and the cell membrane. And our cells do the exact same thing, except that we don't have a cell wall, like I mentioned. So we just have a cell membrane. And that membrane, if it's incapable of separating itself into two separate cells, then it aborts. It actually, well, I mean, there's a variety of different problems with that, but one of the things is that you have a bunch of checkpoints as you go through cell cycle progression, and if anything goes wrong, it immediately cuts the cord and it apoptoses the cell, meaning it kills the cell, it kills itself. So with bacteria, if they're locked in place, they're like, all right, let's go. Okay, all right. But they're not going anywhere. Nothing's happening. Their original army of 1,000 or 10,000 aren't replicating because they've been doused with this antibiotic. Suddenly they can't move. They're just like, well, it's just the 10,000 of us. We're going to have to take on this entire massive army of 5 million. And your immune system's just going to blow right through it. And it's going to be a slaughter. So really, really cool mechanism to stop it from replicating. So you're not killing the bacteria yourself. Like, you're not killing those specific initial bacteria, but you are completely eliminating their ability to essentially call for reinforcements. And then that makes them incredibly weak and susceptible in an environment that's already hunting for them, because your immune cells literally hunt bacteria. So really, really cool. Hopefully you found it just as informative as I did. Obviously I nerd out about this kind of stuff. So hopefully you found it as nerdalicious as I did. And I hope I have the pleasure of speaking with you in the next one. Have a good one, guys. See ya.